Okay, so that was so much of a sort of a place setting, as it were. So what are we going to try to convey today? The first one is, why are we doing metabolomics? Why is it important? I will go through some of the historical developments, the more recent ones, as it were, that bring us to where we are today. And then I'll go over some of the training capabilities, how to do metabolomics, the data analysis, and options for people who are on this call for the future. So the first question, and I think this is a really basic one, what does human life depend on? And of course, that's true of any life. Well, the way we understand it is it depends on the genome that we develop. The genome carries the message as you go through each generation as to how that form of life will develop. So it's built upon the genes in the total genome. Now, the genome per se is a capability. It doesn't actually on itself do anything. There has to be a conversion of that information in the genome into proteins. And it's the proteins as enzymes, as structural proteins, as transporters, as receptors, etc., that actually is life, the form of life that we see as we look at things visually. So it's really important. But all of that, the DNA and having all these proteins expressed is not enough. There has to be fuel to run the ship. So as an example of this, the heart, which is obviously, if your heart stops beating, you end your life. At that point, you have all the genome, you have all the proteins, but the heart stopped beating and you'll be dead. So what is the reserve of the ATP that's needed to keep the heart beating? It's about 10 seconds worth. So metabolism has to be seen as being absolutely essential for life. So when the human genome was actually sequenced and we took a look at it, we were expecting to see 80 to 100,000 genes. And it turned out there was only 20,000. It was in a way, a disappointment. And so the human genome is quite incomplete compared to many other forms of life. So how do we get away with that? Well, it turns out the human is intelligent and it is able to get the forms of the compounds it needs to live by eating, by harvesting what we see around us, whether it be harvesting animals or harvesting plants. So we knew that, and the human is therefore very adaptable. Now the plants have a lot more genes, but the plants have the disadvantage that they can't walk around and add to the food. So they have a lot more genes than we do because they can't move, whereas we can get away with much less genes because we can determine the food we eat. So what is in the metabolome? I think the metabolome, the terminology, the metabolome was first used in 1998. Obviously, people were looking at the metabolome like things I was doing and others were doing before that point. But it's all the small molecules that are found in body fluids, serum, plasma, bile, urine, feces, and a lot of other things that we've measured. And of course, tissues, the heart, the kidney, lungs, brain, etc., when they're available. So the small molecules, they can be made by cells in the body from precursors using the genes and the proteins that are made from the genes. So they can be made as endogenous compounds. Many of them come from the diet. The amino acids that make up the proteins that are in our bodies, only 10 of them can actually be made in the body itself. The other 10 we have to get by eating. And if we don't eat enough of some of those other amino acids, then we're not doing enough for our nutrition. Turns out that the bacteria that are in our guts, which are profound, there's at least the same number of cells, bacterial cells in the gut as there are all cells in the human body. And some people say it's even more. Besides that, for the individual, there are going to be medications that person is taking. When you're young, you're probably not taking much in the way of medications, maybe just some Tylenol from time to time. But then many other synthetic or semi-synthetic substances, which are part of the environment. As we move around different parts of the world, we would expose to compounds in the environment that we didn't know were there, but they appear when we do metabolomics. And we've even moved into the world of metabolism of proteins. I guess before I got into this, I assumed that when a protein was degraded, it was just degraded down to amino acids. But it turns out, in many cases, the proteins are being broken down into smaller peptides. And some of these peptides can have tremendous biological activity and be responsible for disease processes. And until we started doing this, we had no idea that this world exists. So there is a world, I call it peptidome metabolomics, or you could call it metabopeptidomics. But all of this is seen when we examine the metabolome. So is the metabolome relevant to just human research? And could it be relevant to more than that? Well, of course, every aspect of life that surrounds us has a metabolome. And we are consumers. The major agricultural products, the animals and grains and plants and fruits of Africa are all based on a metabolomic underpinning. And therefore, optimizing the utilization of an agricultural product requires a better understanding of the metabolome of that particular material.